So, hi everyone, this is our first lecture of overview series. In this lecture, we will be looking into immunology. So, how are we dividing immunology? As we will be reading immunology as three topics. First is basics, second is applied, and third is clinical. So, I prefer making flowcharts or tree diagrams. It helps us to give an overview of what this topic is telling us. So today we will be looking into basics of immunology. So now we divide the basic of immunology into three parts. First we will categorize it as immunity, second as MHC, which means major histocompatibility complex, and third is immune response. So we will be dividing immunity into two types. First is innate. We will be dividing acquired immunity into both two types, which is active and passive. Now both of these, active and passive, these are categorized as natural and artificial. Both active and passive acquired immunity are categorized as natural and artificial immunity. Second is MHC. We will be talking about it after a few minutes. Let's see about immune response. So immune response, we will be dividing it as HMI, which is humoral mediated immunity, and CMI, which is cell mediated immunity. And second type of categorization is as primary immune response and secondary immune response. So now we'll be looking into immunity. So first we'll be differentiating innate and acquired. Innate versus acquired. Acquired we can also call it as adaptive immunity. So innate means everyone knows that innate is the type of immunity which a child receives before birth, or we can say that at the time of birth the immunity that a child already has, we say that it is an innate type of immunity. So first point is it is present at the time of birth. Okay, and what about acquired is after birth it is seen. Okay, second point is since it is present at the time of birth, it means it is already made before the child is born. So we can say that it is preformed immunity. And what about acquired is okay, preformed immunity. Why do we call it as preformed immunity? Because there is no need of antigen. No need of antigen for the innate immunity to occur. But adaptive, which is after birth, it means it needs antigen. Next is it is non-specific. Since there is no need of antigen, so we can say it is non-specific. And acquired is specific. Fourth point is Innate immunity, it will cause immediate action. But the acquired immunity, it takes some time. So it means we can say that there is some lag period present. Why do we see this lag period present? Because first the antigen will come, antigen entry. After that, antibody will be made, and then there will be action. So because of this, we say that there is a presence of lag period. Fifth point is no memory, or we can say no, it has no history. So we will write no memory of past infection. But here we will have memory, since we will be getting plasma cells and memory cells from the B cells. We'll be talking about it later. So we can say that memory is present. Over here, this point is important, is this is non-specific. Now why do we say that it is non-specific? It means that for any type of antigen, the action is going to be same. Let us suppose that a small antigen, this is small antigen, this is a huge antigen. And in under innate type, what we are saying is that this antibody, this is antibody, let us make it as king. So it will attack it with the same intensity. Let us say it is attacking with intensity capital I. And this is the antigen, this is another antigen, which is, let's say, different antigen. So it will also attack it with the intensity I. But meanwhile, let us say that it is innate immunity. Not sorry, it is an acquired immunity. So in case of acquired immunity, if this is an antigen and this is the another antigen, if the antibody is attacking the small antigen with an intensity of I, then in case of acquired immunity, the intensity will not be I, instead it will be I, or we can say I, anything. But the intensity is not same. What do you mean by intensity? Is that the action that the antibody will take on the antigen in case of innate immunity will be same. It does not change with the antigen. But in case of acquired immunity, the effect of antibody will change with the antigen. So keeping this in mind, let's move further. Now, example of innate immunity, we can say it is phagocytosis. Okay. Now, since we have already discussed that acquired immunity, it is divided into active and passive, and each of these are categorized as natural and artificial. Okay. So let us write it over here. Now this is active, this is passive. Write it as active and passive. Okay. Now active as natural and artificial, again passive as natural and artificial. Now what can be the examples of natural active acquired immunity? It means it can be some sort of infection. Okay. And artificial acquired immunity. Artificial active acquired immunity, it is vaccine. In case of passive, natural, so it can be mother to child, or like it is mother to child, such as antibodies passing from placenta, or we can say it can be via breast milk. Now, artificial passive acquired immunity, it is monoclonal antibodies. There are many types of monoclonal antibodies such as rabies immunoglobulin, tetanus immunoglobulin, etc. Done. So we have completed our first thing, which is immunity, its types, and with some examples and comparing innate versus acquired. Now we will discuss about MHC. So this is an important topic. MHC is, we see it as major histocompatibility complex. Now, what we have to remember over here is, first let us see a story. Let us suppose an antigen enters our body. Entry. The first thing that our body will do is phagocytosis. Why? Because innate immunity is the first line of defense. If we have to remember that if this fails, then the acquired immunity comes in action. Directly, acquired immunity will never come in action. So, antigen entry, phagocytosis, it means if antigen survived, there will be some messenger who will go and talk to our acquired immune system. That sir, there is someone who came into our body and he survived, and our innate system and our innate immune system couldn't win against the foreign particle. So now you have to take in charge. We can say that this is local police, innate is the local police, and acquired is the military. So when the local police cannot handle the terrorist, then there comes the acquired or the military. So similar is the case that when the antigen survives phagocytosis, there is going to be some messenger. Some messenger will go and talk to the acquired immune system that, sir, go now, you have to take in charge and kill the foreign particle. This messenger is nothing but MHC, major histocompatibility complex. So we can say that MS is nothing but a messenger. Okay, so this is the story. Now, start. So MHC is the messenger which comes in action when the innate immune system fails. Okay, done. Now, MHC, there are two types of MHC or the major histocompatibility complex. This is MHC1 and MHC2. So now I am presenting the diagrams of MHC1 and MHC2 molecule. Now, in MHC1, what we can see is there is 3 alpha molecules. Okay, this region that we can look at over here is antigen presenting site, or we can say antigen binding unit. This region is antigen binding unit. Now what we can see here is First point that we need to remember in case of MSc class 1 molecule, we will be differentiating both the classes. In MSc class 1 molecule, what we can see is there the antigen binding unit is present between one unit, means between only alpha molecules. Second point is there is only single attachment, so one unit, one attachment for MHC1 molecule. Now for MHC2 molecule, this is again the antigen binding site. The antigen binding site or the antigen binding unit is, now this is one alpha unit, sorry, like this is alpha unit and this is beta unit. So the antigen binding unit is present between, we can say, between two units or two different types of unit. So two units for an MHC2 complex. Here we can see there is two attachment. So two attachment, two units for MHC2 complex or molecule. Now the important line. This line must be memorized or by hearted, just cram it, but this is a very important point in immune system or the complete immunology, which is, now first let us see the location. Now the MHC1 molecule will be present in all nucleated cell. It means RBC. And MHC2 molecule, it will be present in professional antigen presenting cell. It's nothing but APCs. Now these are three types: B cells, macrophages, dendritic cells. Now, since we know that B cells, macrophages, dendritic cells, these are also nucleated cells. So will these not have MHC1? No. These will have both MHC1 and 2 will be present, but only MHC2 will be activated. Only MHC2 will be activated or active. So all nucleated cells will have MHC1, and B cells, macrophages and dendritic cells, which is the antigen presenting cells, will have both 1 and 2, but only 2 will be active. Okay, now since we said that, okay, now the important point that we talked about, that this is the most important point in the complete immunology, is not yet over. Now what we need to look at is Since these are messengers, as I said, that MHC will take the information. Now, who is the information? It is the antigen, which is the information. So, MHC1 has antigen binding unit. It means that it will carry antigen. Same goes with MSC class 2. So now we need to remember that MHC1 will give antigen to whom and MHC2 will give antigen to whom. So MHC1 will give antigen to T cell receptor of CD8, which is also known as cytotoxic T cell. MSC1 will give present antigen to CD8, and MHC2 will present the antigen to T cell receptor of CD4, which is helper T cells. So how we can remember is, remember the number 8. So 1 into dash equals 8 over here. This is MSC2. So 2 into dash equals 8. So we will be writing 8 over here. 1 into 8 gives us 8 and 2 into 4 gives us 8. So this 8 represents CD8 and this 4 represents CD4. This thing is the most important thing in the immunology. So now we will be looking into a new story. Over here, what we need to look at is, now this story is very important. So let us suppose that an antigen that entered our body, and the first thing that we know is our innate immunity comes into action, which is nothing but phagocytosis. Okay, now phagocytosis, let us suppose if it is successful, it means our body is happy. But if it couldn't kill the antigen, then MHC complex will take place. Now, what will happen is, the MHC complex, as we know that it will take the antigen, the MHC complex will take the antigen and it will go towards acquired immune system. Now, since we know that phagocytosis is done by macrophages, and macrophage, now it has both MHC1 and 2, but we know that only MHC2 will be active. So now what will happen is, antigen in the macrophage is nucleated, it has MHC2, so the antigen will be bounded to MHC2. Okay, now what do we know that I said about the number 8? So we are talking about MHC2. So 2 into dash gives us 8. It is the 4. 4 plus 4 means it means CD4. So antigen, it will bind to MSC2. And where will it bind? It will bind to the antigen binding site. So antigen will bind in the antigen binding site of MSC2 molecule over here, and it will go to CD4. Okay. Now we will consider the CD4 as king and the MHC2 complex as a soldier. Okay. Now, in order to meet the king, the soldier will have to show a gate pass that I am not your enemy, I am a member of your kingdom, of your army. So, after looking at the gate pass, the king will allow the soldier to enter into his palace and meet him. Okay. So, what is the story till now? That there is a soldier who will have to show a gate pass to meet the king. Okay. Now, after meeting with the king, the soldier will say that, sir, there is an issue and we have some intruder in our kingdom. We couldn't deal with it. So now we need your help to get rid of the invader. Okay. Now the king is very powerful. He will think that, okay, our soldier, our army couldn't do anything. So now king will use his own power. So now the king has its own military. Okay, so now the king has his own military. Now the military will select its one and only one best soldier. This soldier is nothing but B cell. Now let us clear the story. What do we mean by all these things? Now this soldier is MHC2. The gate pass. The gate pass is interleukin 1. It means the soldier gave the gate pass. It means the MHC2 will secrete interleukin 1. The interleukin 1 activates the king, which is CD4. Now, after activation of CD4, CD4 will release military. So, military is here interleukin 4. CD4 releases interleukin 4. Now, interleukin 4 means the military selects its best one soldier. It means interleukin 4 will select a B cell. This process, it will select a B cell. This B cell, this particular B cell will undergo multiplication. After multiplication, it will form memory cell and plasma cell. This plasma cell, it will secrete antibodies. This antibody will go and attack on the antigen, neutralizing it. Now, let us revise the story. There is a foreign antigen. Let us revise the story first. So let us think of terrorist. Terrorist came. Our local police, or we can say soldier, couldn't do anything. So they went to the king. But to meet the king, they had a gate pass. By looking at the gate pass, they met with the king. They met the king. The king, on looking at the condition, he selected his military. Military people selected one best soldier. This is our story. Now, terrorist over here is antigen. Local police is MHC2 complex present in macrophages, which couldn't kill the bacteria or the antigen via phagocytosis. Now, MHC2 will release interleukin 1, which is the gate pass. The king over here is CD4. Interleukin 1 activates CD4, and activated CD4 will release interleukin 4. CD4 will release interleukin 4. Interleukin 4 does select one B cell. This B cell undergoes multiplication, producing memory cell and plasma cell. This plasma cell produces antibody. Now, the antigen which is carried by MSC2 complex to CD4, this process is known as presenting of antigen to CD4. This is known as antigen processing. It means taking the antigen to CD4. It is antigen processing. So antigen processing means antigen in the MSC2 complex is moving towards CD4, but interleukin 1 is not yet released. After interleukin 1 is released, CD4 gets activated, and this process is known as antigen presentation. So the process of taking antigen towards CD4 is antigen processing, but the process of activating CD4 is antigen presentation. Now, since the CD4 gets activated, we know that it will secrete interleukin 4, and interleukin 4 selects one B cell. This process of selection of one B cell is known as clonal selection. And the multiplication is known as clonal proliferation. Stop. So this is our whole story. Since there were no cells involved, Since there are no cells involved, so this complete process occurs in case of humoral mediated immunity. So what is the story? There is a terrorist, and local police couldn't fight against it. So what they did was they were going to the king, that they were presenting their case. So when they were going, the process of going is known as antigen processing. So terrorist, local police, fight, couldn't win, going to the king. The process of going to the king is antigen processing. The gate pass. After looking at the gate pass, the king gets alert. It gets activated. So this process is known as antigen presentation. After presenting, the king talks to its military that now you have to do something. Then what military does is it selects its best soldier. This is the story. Now, what actually happens is the antigen, it binds with MHC2 complex Or we can say that the MSC2 complex takes the antigen towards the king. The process of going towards the king is known as antigen processing. And after reaching king means CD4. Now the MSC2 is going towards CD4 is antigen processing, and release of interleukin 1 which activates CD4 is antigen presentation. CD4 on activation releases interleukin 4 which does clonal selection, which means selection of one B cell, and this one B cell undergoes proliferation, giving us as memory cells and plasma cells. This plasma cells releases antibodies. This complete process is seen in case of viral mediated immunity. Now we will look into cell mediated immunity. Again, we have a terrorist. Let us look into the story first. We have a terrorist. Now this terrorist came into my house. Silently, without my prior information or knowledge, this terrorist entered my house and he was sitting in my room. As I entered my room, I saw him and I asked him, "Who are you?" This terrorist revealed that it was a virus. Now, on seeing the virus, I got scared and I went to the top of my house and I started shouting help until I completed the last alphabet P. The local police, they came and they said, "What is the problem?" Then I said that there is a virus in my house. Stop. Start. This local police is nothing but MHC1. Since it is a cell, the house, terrorist came into my house, so the house is cell, and every cell which is nucleated has MHC1. So the MHC1 will stop. Start. The MHC1 will interact with virus particle. And after interacting, it gets activated. Now the activated viral particle, the activated MHC1 complex will release granzymes and perforins. It will release granzymes and perforins which will cause perforations in the cell, ultimately leading to cell lysis. It means that my house is destroyed by the local police because of the virus. It is not killing the virus, it is completely destroying the cell in case of cell mediated immunity. Now, here I mentioned that the MHC1 is activated. I was mistaken. The MHC1 will present the activated stop. Now the activated CD8, since MHC1 will transfer the antigen to CD8, activated it will release granzymes and perforins. Stop. Start. Now, let us suppose that the virus, it did not enter my house. It saw that this house is there, but it is not so beautiful, so I will not go there. This virus saw this beautiful macrophage over here. Here is the macrophage. So this virus entered the macrophage. Now this macrophage will have MHC2. It means it will be presenting to CD4. Okay. So again, it did phagocytosis, same thing. Virus entry, virus nothing but viral antigen. So antigen entry, phagocytosis fails, then MHC. Since it is macrophage, we will consider MHC2. MHC2 will take the viral antigen to CD4. Now again, stop. Now the same thing comes. Start. So MHC2 will carry the antigen towards CD4 is known as antigen processing, but after releasing of interleukin 1, the CD4 gets activated. It is known as antigen presentation. Since this is a viral antigen, in case of viral antigen, CD4 will release interleukin 2. But in case of normal antigen, the CD4 releases interleukin 4. We need to remember this thing. Why? Okay, see. CD4 is releasing interleukin 2 in case of viral antigen. This interleukin 2, this will again activate, or it has the ability to activate CD8, and after the CD8 gets activated, it will release granzymes and perforins, ultimately causing cell lysis. It means that CD4, why did we say that CD4 was a king over here? Where is our king? Yeah, we mentioned that CD4 is the king. This is the reason why CD4 is the king, because it can release interleukin 2 as well as interleukin 4. By release of interleukin 2, it can activate CD8, which can, and CD8 is involved in cell mediated immunity, and via interleukin 4 it is involved in humoral mediated immunity. Since it is having, or it plays a major role, it can play a role in both humoral mediated immunity and cell mediated immunity. We called it as the king. Stop. Start. So this was the story for cell mediated and humoral mediated immunity. Now coming to our primary flowchart, what we have done is we have done our MHC complex, as well as under the immune response we have done humoral and cell mediated immunity. Now coming to primary immune response and secondary immune response. So now we will be looking into primary immune response and secondary immune response. So again, we will be looking into a story. This story is not my story. My teacher presented the story and it was a pretty nice story, so I am telling the story over here. So in this story, there's a boy, a naughty boy, a mischievous boy, who one day In a microbiology class, he made a whistling sound. It means कि एक लड़का सीटी मार रहा था क्लास में. So the teacher got very angry. He said the student, go meet the HOD sir. The student went to the HOD sir, and in the room, sir said him to sit down and asked a couple of questions. Who are you? What is your badge? His name. And in the end, he asked a very important question, which is, why did he come here? So the student says that he made the whistling sound. So by listening this, the HOD teacher got very angry. He scolded, and in the end, the teacher said the student to write an apology letter. The student, he wrote the apology letter. This blah blah blah, I will not do this. I am very sorry, I am feeling guilty. From next time this will happen, I am responsible for everything, blah blah blah. The student wrote and he went away. Again, after 2-3 days, in the same microbiology class, the student did again the whistling sound. The faculty of microbiology, he got very angry. He said that go and meet the HOD sir again. So this time when he met the HOD sir, he will not ask those questions. Who are you? What is your badge? He will simply ask one question. Did you make the whistling sound again? It means it was in the memory of HOD sir that this person made the whistling sound before. And the student will say that yes, I made the whistling sound. By listening this, the HOD sir will give him a serious punishment, suspension or whatever. So the first time when the student made the whistling sound, it means that there were a couple of questions, but there were no action taken, no serious action taken. It means the first time he made the whistling sound, there was a long duration. It means that there were many couple of questions, but there was a very low severity of action. But the second time when he made the whistling sound, in that case there were very less questions, but the action was very severe. It means the first time when the antigen enters our body, the duration will be prolonged. It means that It means that the action of primary immune response will be slow. Teacher is asking couple of questions. So in case of primary immune response, it is slow action and very less severe punishment. It means sluggish. And it is very short-lived. Prolonged memory. And in case of secondary immune response, the action is very fast. Fast action, prompt action. Here, when the student did the whistling sound second time, the teacher gave very serious punishment. It means powerful action. We can also make a graph for this. Let me make the graph. So Um, as you can see here is, this is the primary response, primary immune response, and this is the secondary immune response. Here this is the time on x-axis and this is antibody concentration on y-axis. It means that we can see here the antibody concentration is very less, but the time taken is so much. In this much time taken, the antibody concentration is less. Meanwhile, in case of secondary immune response, the time taken is very less. And the concentration of antibody is also very high. Example of primary immune response in the form of vaccine, and secondary is nothing but we can say booster dose. This is all about primary immune response and secondary immune response. So looking into our main flowchart, we have successfully done the basics of immunology. In the next lecture, we will be looking into applied aspects of immunology. That's it. Thank you.